So my name's Martyn-Dade Roberson. I'm a reader in design computation at the School of Architecture, Planning and Landscape at Newcastle University. And I'm unusual in that I have, I guess, three fields of interest, really. My first, it was in architecture, and I teach architecture now. My second, where I did my research and PhD, was in computation and architecture. But my more recent interest is in synthetic biology. In fact, I have a master's degree that was done as part of a sabbatical 3 years ago in microbial synthetic biology. The work that I'm going to show you today is about that, really, about that, some initial explorations and some research that I've been doing in my group. And the things that we've done since the last meeting, there's an expanded numbers, but some of you will remember me from the first meeting, those of you who were there, and we've developed a little bit since then. So I run a research group now, which is really a cross between a scientific research group, an engineering research group, and a design research group. Our interest is in engineering bacteria-based systems to make new materials, specifically for construction, but we have lots of interest in all kinds of different aspects of the work that we do. And the idea is that we have a design practice where we can go from the molecular level, so we're looking at individual cells and the things that are happening with individual cells, and in fact even individual genes within cells, right up to the building scale. And that's the ambition that I've set my team. Now the problem is that as an architect I have far too many images of things down electron microscopes and not enough pavilions, but we're working to try to scale up, and I think this is something that there are a few people in this room who recognize the challenge. So I've now, since I presented last time, my research group has grown, and so the homogeneous nature of their attire should not give you the sense that this is a homogeneous group, far from it. So from left to right, we go from civil engineering on the one side, so Javier, who's a PhD, he's just finishing his PhD as a civil engineer, specializing in materials and materials testing, right the way through to Beata, who's a microbiologist who works in my group, and then wedged in the middle are my architecture PhD students. Wedged in the middle. Architects tend to be wedged in the middle of these sorts of things, and this is where they sit from all kinds of places. So I'm going around collecting some of the best master students that I can find from different places, including IAC and University College London and other places. And some of you might well recognize one or two faces in that. So we've built, and then we have a much broader team. So I'm blessed to be in a university that has a whole range of different departments, bioscience, computing science, and so on. And we work across with many different groups, and we have members who are kind of affiliate members of our group all over the place. The big change for me is that I now have access to a lab. In fact, I have my own lab. So it's a Cat 1 lab. It is humble, as you can see, and really badly poor resolution, but it usually looks better than that in real life. But broadly speaking, even though we've got our own space that we can kind of organize, we can start thinking about the biology lab not only as a biology lab, but a space to do multiple different things. And so in our lab, we've got, I put these lines like they actually mean anything. What happens is that on some days we have our 3D printers out, then on some days we have to put our 3D printers on shelves because we have agar plates out, and then on other days we pull out the microscopes. But what we're trying to do is to create something more akin to a design studio actually than a biology lab, but within the same constraints, so that we can work in a sterile environment, we can grow things and we can do things properly. And we're hoping that over time we'll expand that. We're already finding that it's overused, and we have a constant stream of students actually using the facility as well. What I'm going to do is just talk very briefly about a few of the projects. I'm going to give you only a couple of slides on each one. And these are really designed as little tasters, because all of these are things that I want to talk to people about, I want to have collaborators for, and it might spark some interest in the team. The big project for us is called Thinking Soils, and I presented this at the first LASG meeting. As a result of that meeting, actually, we had some buy-in from LASG to help put the next project together. And the project became Thinking Soils. So we had a project that was only 18 months. Now this is 3 years, multiple collaborators, and this is the big one, really, that pays and runs my lab. So the idea of Thinking Soils is it's a simple idea but a complicated problem. So what would happen if we wanted to build a building, and rather than digging a foundation plate, filling it with reinforced concretes and building our building on top of it, we got the soil to respond, to remodel itself in response to load? How might we do that? What might the possibilities be there? And in my naivety, I thought, well, perhaps the bacteria in the soil could act as pressure sensors to us. Perhaps they could detect mechanical changes and then synthesize a material in response. That was a really naive architectural question to ask, but it turns out to have some interesting sub-questions. And so what we did was to organize a research group into multiple scales. And you often find that different disciplines work in multiple scales, work in specific scale domains. So molecular biologists are interested in the things that happen within the cell, and the microbiologists are interested in the cell and its interaction with its environment. And so we set up a team to look at each of the scales, right from looking for sensors, genetic sensors that might be responsive to mechanical changes in their environment, right the way through microbiology to look at how the cells interact in large communities within soil and soil-like materials. In this case, we're using jellies to grow three-dimensional colonies of bacteria, right to looking at the mechanical changes that happen in soil and soil-like environments. And we're putting all those things together almost piecemeal, almost independently, but then trying to pull all that knowledge together, in a single system. So we've written our first papers looking at some of these these scale questions, and just we're beginning to get to the point now where we can put some of this knowledge together in one place. And the image I'm showing you here is a computational model that we've been working on for the last 3 years that takes the data from our models about what's happening within individual genes within the cell and data from large-scale analysis that we have of forces within the material and puts them together. Now, it would take me quite a while to explain what this model is, but if you wanted me to explain that, I can talk to you about it and show you some proper demos of the software in operation afterwards. But this is really the first step to thinking about these multiple scale problems, and the results are really fascinating. This is effectively sculpting with genes, which is just a really interesting concept and one that I'd like to develop more of. Another project I presented last time, very tentatively, was the idea of using bacteria spores to create hygromorphic materials, so materials that can change their shape in response to water. We did some earlier pilot studies with our students. We've been trying for a year, somewhat unsuccessfully, to make our first hygromorphic materials. Just to say that this is not a new concept, so those of you who know your current architectural and material thinking will know that wood-based hygromorphs have existed for quite some time in architectural discourse. And actually bacteria spore-based hygromorphs, this is where the bacteria spores, the seed form of the bacteria, are mixed in a solution and then applied to a layer, such that as the material gets exposed to water, the spores expand, and as they dry, they contract, can create these cantilever, these tiny little cantilever structures that will move in response to changing humidity. And there has been design interest from places like the Royal Colleges of Arch and MIT in making something with these materials. Our breakthrough really came over the summer when we had an undergraduate student who came into our lab, realized this is an undergraduate, a second-year undergraduate student, realized that we were doing it all completely wrong, decided to do everything from scratch, made her own humidity and dry chamber, worked with a different set of materials, this time latex, which is much more amenable to the kind of work that we wanted to do, and demonstrated a process where she could get the spores to self-assemble on the material and then adapt and change to humidity predictably. So she could program the material that as she added on layers of spores, I should stop doing that. The material would flex and change more and less. And then she connected that to a system of what she called Octave active origami, making origami structures that were able to change in response to the humidity of their environment. And I'm hoping that she's going to start a PhD with us in the summer. I think there are lots and lots of places we could go with this stuff. Oops, so. I'm going to skip through a couple of slides now because I want to get to the end, and I've only got a minute left according to my thing. But I've spoken to people. One of the challenges that we've had is that often we don't scale very well. There's a real interest, I know, from some people in this community in using mycelium. We just started that process ourselves. We're getting our students to make lots of mycelium-based materials, and there's a real fun in experimenting with all the different ways that you can grow this material. And some really interesting research questions. We're very interested in the idea of making multifunctional materials using mycelium, but growing them in one go. So thinking about biohybrid materials that can be grown in one shot, but have lots of different material functional properties through their section. And we're interested also in their interaction with other materials, particularly timber and other materials where they might act as an adhesive. And so there are certain things coming on that. And we're in the final round of an application with NASA, actually, to look at this material and how it might be used for colonizing other planets. I could talk to you about test tube timber, but I'm not going to. But if you're interested in growing wood in a lab, then talk to me about that later. Just finally, I wanted to, this is a kind of a call really, and this is probably the best audience I have to make this plea, but as we've grown, we had an opportunity this year to apply for a huge grant, actually about £10 million, to create a new centre for biotechnology in the built environment in Newcastle. We find out in this month, any day actually, whether we've been successful or not, and at the current plan we have 50-50 chance of getting it. If we do get it, then we will be expanding very rapidly. In fact, we'll have 15 new tenure track positions, we'll have a couple of new professors starting, and a whole host of postdocs and PhDs. If you are interested in that, if you have people who, you might be thinking of a new job, Newcastle's a beautiful place to live, or you're perhaps coming up to looking for tenure positions or whatever, then please do let me know, because we need to hire very quickly if we get this grant. And this would be really, this community is where a lot of those people will come from. At the heart of this grant is a new idea, and what we want to build is an experimental house, a complete house that we grow and that that we can use as a test rig for all our technologies and ideas. We're calling it the OHM, and it will be a combination of a home, but also the other things that we think of when we're thinking about biology. So a conscious design of the building's genome, the proteome of the microbial environments, the metabolome, and the microbial biome as well. Thinking about all these things together in one go as we design this thing. If we get the funding, this will be the first thing that we produce, and will be far the most advanced, the exciting projects I'm involved in. The other thing is that I've just been commissioned to develop a new Routledge series on biodesign, for which I'll write the first book, or I will if I ever actually get down to sitting down and writing it. This is not the cover, by the way. They will get a proper designer to design the cover for me. But if people are interested, it's going to be 30,000 words, and we're looking for authors who might contribute useful, really interesting books that are about the right size for designers who don't want to commit to doing a whole monograph, but want an opportunity to explore some of their design work or their theories in more detail. And I have information on how to apply to do one of those, and as the commissioning editor, I'm really keen to hear interesting ideas, and I've spoken to one or two people about this already. The other thing is just to finish to say that we're also hiring. So regardless of whether the big project comes along or not, I really, really do need people in my lab. So my lab's going really quickly. I need a computational design specialist really badly. If you can start tomorrow, it would be great. Who can build computational models that look at biology and, oops, doing that again. Who can mix between our biological modeling and our physical modeling and build new ways of visualizing that stuff. So again, if you're interested, then come and let me know. The advert hasn't gone live yet, but is about to very shortly. So yes, please get in touch if you want to write a book or do anything or that. And also our website is down at the bottom if you want more information. Thank you.